Good evening. This is Maly 8 News, and I'm your host, not Ron Burgundy. In our first segment, we'll be joined by Cindy, who will tell us more about experiments she's using to study CRISPR-Cas9. Hi, Cindy. How's it going?
Good.
Tell us what you're up to.
I'm just prepping some samples, load onto the sequencers for my experiment.
Look at all those sequencers. What information will they give you?
So I'm looking at how CRISPR-Cas9 edits at on and off target sites within a cell. And to do that we use DNA sequencing, specifically Illumina next generation sequencing, to kind of read out of our experiments.
Very cool. What other exciting projects are you working on?
Yeah, so I'm working on several different protein engineering projects where we're making some off switches for our chemically inducible Cas9, and also some base editors and prime editors in a chemically inducible version of those.
So, you're from New Zealand. Will you solve the age-old mystery for folks at home? Do the toilets actually flush backwards and land down under?
So it's hard to tell because when you flush the toilet it goes so fast. But there are some cool YouTube videos where they like stabilize the water and all that stuff, and it does go the other way around.
Mystery solved. You cracked the case. Thanks, Cindy. Ooh, you know what that sound means. The deep mutational scanning data is in, and it's time to check in with Jessica. For more, this is Yeast Watch 2021.
Hey, not Ron. Our DMS results are just in. Up here, what we're seeing, as expected, most of them are loss of function, but there are a couple right around here that surprisingly are gain of function. Could this be an unknown regulatory mechanism? Let's look at the individual curves and find out. So, what we're seeing here, they're just coming in. Empty vector is growing very fast, as we would expect. That's good, that's good. The wild type, a little bit more toxic, also good. But this E to T down here, hardly growing at all, but we'll have to wait and see if it takes off. See you in a couple minutes.
Thanks, Jessica. In our next segment, we'll visit Ethan, who's using chemical tools to study intercellular target engagement using microscopy. Wow, look at those cells. How's it going, Ethan?
Hey, how's it going, not Ron?
Tell us more about what you're working on.
I'm working on cleaning up some antibody for a microscopy reaction that I'm going to be running later.
What are you going to measure?
I'm going to be measuring how inhibitors engage with their targets within cancer cells.
Wow, what a powerful technique. Keep it classy, Ethan.
Hey, you too, Ron Burgundy.
Next, we'll check in with Dan, a first-year graduate student with a passion for proteolysis targeting chimeras, or PROTACs for short. For more on that, here's Dan. Hey, what's up, Dan?
Oh, hey Zach, how are ya?
Doing well, how are you?
I'm doing well.
So, I hear you make these small, teeny tiny little molecules, and then what do you do with them?
Yeah, so I have them in a DMSO solution, and I'm gonna go ahead and treat the cells that are in media, and then I can go ahead and measure protein concentrations afterward, and we can see if they actually do any degradation.
Very cool. So, I have to ask the question that's on everyone's mind. Do you know if the mass spec downstairs is running?
I think so.
Well then, you better go catch it, Dan. Ooh, you know what that sound means. It's time for another update on Yeast Watch. Here's Jessica.
Thanks, not Ron. These curves are looking really interesting. So you can see empty vector has essentially topped out here, stopped growing. But this E to T variant is still looking really toxic, meaning our enzyme is really active. But what does this really tell us? Well, it gives us information about protein-protein interactions, allosteric regulation, all sorts of stuff. But to really figure out what's going on here, we're gonna have to do a little bit more digging. Back to you, Ron.
While I have you here, I wanted to ask, would you rather drink a Starbucks coffee or a beer from Fremont Brewing every day for the rest of your life?
That's a tricky question, not Ron. I'm gonna have to go with the beer from Fremont Brewing. Pretty tasty, especially on a nice warm Seattle summer day.
Thanks, Jessica. In our final segment, we join Gayani, whose projects include making small molecule inhibitors for prostate cancer. Hi, Gayani.
Hi, Zach.
How are you?
I'm doing well. I just came back from a prostate cancer meeting.
Now, that's a big collaborative project, isn't it?
It is. It is a big collaborative project. There's like groups from UW Medicine, Fred Hutch, so a lot of scientists there.
Is that pretty common for projects in Dusty's lab?
Yes, as far as I know he has been having collaborative projects forever. And those have been super successful and also good for the students because, you get to expose to people from different fields, like there are biologists, biochemists, protein engineers, doctors, bioinformaticians, like lots of research exposure there.
Sounds very rich. Now, some years ago, you were also one of Dusty's first graduate students. Can you tell us one of Dusty's characteristics you remember being important for your success?
The best quality I would say is that he's always very approachable. So say it is something related to your research, your academic work, or your personal thing that you want to talk to, you can always rely on him to be understanding and be supportive. So during grad life, I think that is very important.
Thank you so much for checking in with us. See you later.
Thank you, Zach.
That's all the news we have time for tonight, folks. I look forward to sharing my research with you when we meet at the poster sessions or during the other recruitment events. See you there.